talk about predation a little bit. And no celebrations. We will not be celebrating predation. But I did find a predation cartoon that has fish in it. You know what sound he's making, right, with that tuba? Donna. Although it's actually, I think it's a string instrument that makes it in the actual movie. Is it a cello? I've played the cello. Donna. So I should do that. I used to play the blues though. Dun, 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 dun. So predators. But what are the great white sharks of the stream, you might wonder? Well, these are pretty scary. If you're a fish, these would be scary. And predation, of course, is one of the ways that vertebrates can interact in food webs. Not the only way, as we talked. But as we're discussing food webs, we need to keep the terrestrial realm. These are kind of scary, aren't they? The longer I look, those beaks. Ew. So we talked about HSS fretwell trophic theory already, right? And this idea that in some cases, we can have pretty strong top-down effects, right? So HSS fretwell trophic theory basically says that you start on the top of the food web and assume that it's not predator limited, and then you move on down, and you can figure out which trophic levels are limited by either predation or the level below it. But bottom-up limitation can happen as well, right? In other words, we could talk about producers, and so bottom-up is essentially the case if you're not limited by what's above you, right? So this is maybe a framework. So in some ways, predation is like a special case. Well, in some ways, herbivory is like a special case of predation. They're just predators on plants, essentially, right? So keeping this framework in mind, and we talked about Mary Powers' work, which showed that despite the importance of disturbance on stream ecosystems, these biotic interactions are important in certain contexts, right? These grazers are important, both in Panama and in Oklahoma. Did we talk about Allen's paradox? Maybe did we talk? So I had this weird feeling that we had, but I searched all the PowerPoints, didn't find it, and I'm like, did I put it on a PowerPoint that I didn't save to my hard drive? Anyway, so I think we did mention this study briefly, where this author, actually the author of your textbook, one of the authors, conducted this experiment where they reduced trout density to a quarter of what it normally was, and apparently had no impact on the invertebrates. So looks at that and says, you know, there's probably not a whole lot of biotic limitation going on in this ecosystem. It's probably more abiotically driven, right? Well, even before then, there was this concept called Allen's paradox. And Allen's paradox was that when you look at the biomass of trout in these streams, the benthic macroinvertebrate production did not seem to be enough to support them. Like, there didn't seem to be enough for these trout to eat in the streams. But not only were the trout able to survive, there was actually energy exported downstream with the macroinvertebrate drift. So there was this puzzle. This is a paper actually published back in the 50s. And there's this mystery, like where is the energy coming from in these streams to support this high level biomass of predators? So Alexander Urine in New Zealand took a stab at this. And the stream he actually worked in was a tributary to the stream in New Zealand. He wasn't there when they were filming Lord of the Rings or anything cool like that. They might have been filming another movie, I don't know. Everybody likes to film movies in New Zealand. So went to a small stream and said, all right, let's do some detailed calculations about where do these predators get their energy from, how much energy is in these different components. Let's really kind of break down the energy budget of the stream. And some really intense calculations. So he did this interesting thing actually, where he sampled invertebrates using these what did he call them? I forget what he called them. They were essentially like buckets full of substrate that he put down in the stream. right? Like buckets full of substrate with holes drilled in them so invertebrates could get into them. Then he would take that bucket out of the substrate. Well, first he'd take a server sampler on top of that substrate, see how many invertebrates he got, then compare it to how many were actually there in the whole bucket. So he got an idea of how efficient the server sampler was. He also was able to determine how many invertebrates weren't captured by the server sampler. Now, why might invertebrates not be collected by a server sampler? Because they stuck on the rocks? And, how deep did our hands go in that substrate? I don't know, a couple of inches maybe? Well, if you got a lot of bedrock and stuff, it's probably not going to have a big deal. But if you've got a lot of gravel, there's the potential for invertebrates to be down deep in that substrate too. So anyway, so this p-value was the difference, was whether or not there was a significant difference between the invertebrate abundance estimates in the whole substrate versus in the server sampler. And then he went through and he dried and weighed lots of invertebrates to get an idea of how much energy they actually contained, and then multiplied that across all the site just to see where the energy was. So this is what he found. It turned out that this was his estimate of benthic secondary production. And I'm sorry, I don't have the units here. Actually, he measured it in ash-free dry mass. So you take the organic matter, you dry it, you burn it, you can determine how much organic mass was in them, right? How much energy was. So lo and behold, the same held true. The trout demand for energy was greater than what was being produced by the benthic invertebrates. So Allen's paradox still kind of there, like what's going on? And trout production was down here. Why is trout production so much lower than trout demand? Larger, yeah. And when we look at, the amount of, this is the amount of energy that the trout are taking in. This is the amount of energy that the trout are essentially storing. So this is like trout growth, right? This is how much energy they're taking in. And so the good old pyramid of numbers, right? There's this much energy that you needed for the trout, but of course that did not get turned into trout production to move on up the food web. So how was this paradox resolved? What might have been going on? Any thoughts? Mm-hmm. So like the biofilms, the microbial loop in those organisms. So that energy would probably get to the trout through macroinvertebrates. So like the myofauna, you're right. So the myofauna connects to the energy in the microbial loop. Then that myofauna will be consumed by other macros in it, so it'll move on. So you would expect that it would be captured kind of in that energy flow, but he didn't actually measure the myofauna. He sort of, because I think he assumed that myofauna were too small for trout to consume directly. So the idea is by measuring the macroinvertebrates, they were consuming them. Right. That's a good. Maybe the trout are benefiting from some microbial production. That's a good question. Yeah, I haven't read much about that. And so of course they measured. So like the shredders would be included into here, but they got some data on other things. So that's one possibility. What else might have been going on? What else did trout eat? Very, very small children. So it could be terrestrial invertebrates. Yeah, could be terrestrial invertebrates. Yeah, so it turned out that in order to balance the energy budget, a couple different things. So it was terrestrial invertebrate input, that was important. They measured that by putting drift nets in the stream and capturing the terrestrial invertebrates around the top of the water. Also, cannibalism. Trout eat other trout. Trout tastes delicious. You guys know how expensive it is in the supermarket. If you're a trout, are you going to eat some trout? Sure, why not, right? No, sorry, that was weird. And then the final piece is what's called hyporeic production. So breaking that word down, hypo means what? Low, yeah. So like, is that what you said, low? I love these masks. Somebody says something and you really have no idea what they said. Exactly. And then you say what you think they said, which may not have been what they said. Yeah, sure. Like a hypodermic needle goes like under the skin, right? So it's like low, right? So yeah, so the hyporeic, where is it? Oh, there it is. It's actually like below the river water. So it's that layer of substrate like down deep. Turns out, wouldn't you know, invertebrates live there too. It's apparently nice this time of year. So that's production that you're not going to get from a Cerber sampler, right? So 10 centimeters, how many inches is 10 centimeters? It's maybe what, like that? What's 10 centimeters? I'm having a weird time visualizing. I can visualize a meter. All right, it's a tenth of a meter, which is like 3.9 inches, right? Yeah. So it's not even that deep. Like that maybe? Something like that. So, we definitely didn't go deeper than that with our Cerber samples. But so there are apparently invertebrates living deeper than that. And sometimes they come up and they get eaten. So, so the only way to balance this budget to come up with enough energy that fueled the trout was to include these things. So when they summed up, this is all the total available prey, right? So these are the macroinvertebrates. So this is what's available. These are the sums over here. So this is what's available. Surficial macroinvertebrates, those are the ones on the surface. Makes sense. This is hyporeic production. It's what, I don't know, maybe a fifth or a sixth of the stuff you catch with a Cerber sampler. Sounds like something you'd order as an appetizer when you're out at dinner. I'd like the Cerber sampler. Excellent. Will you like to share that? Would you need many plates, or do you just want? Sorry. And then terrestrial macroinvertebrates, at least in this stream, were not much compared to the surface bugs. And then potential cannibalism based on what they study. So a little bit there. They don't eat each other a lot, just sometimes. Just when you're really hungry. You guys saw the movie Alive. James, you're asking me to go back and read their methods on how they estimated cannibalism, which I could do. I could do that. You don't make me do that. No, I will. They don't make these things up, but I'll look. I can look that up if you like. Maybe while we're transitioning to our paper. These are real numbers. They didn't just be like, let's make the shapes equal. Let's see. Or maybe they did, but no, they said. Yeah, but so check it out, right? This, when you sum them all up, that's the energy that's total available prey. And then when they estimated how much the trout had to eat, and they did that based on metabolic efficiency, right? So you look at given size of trout, what their metabolism is, how much energy they need. This is how much the trout demanded. They're very demanding, those trout. They're pretty close to equal. It's basically saying in streams, if you've got trout hanging around, that's where all the energy's going. There's not much extra. What does this suggest about the potential role of competition in streams as it relates to trout? Do you think competition is high among trout or low? Yeah. Is competition going to be high when there's plenty of resources to go around or when there's scarce? When there's scarce. And it looks like that stream produces just enough energy to support the amount of trout in it. Now, when do you think that competition happens? When in a trout's life is that competition likely to happen? The life of a trout. So, trout hatch out of eggs that are buried in the stream. They make nests out of gravel. And when they emerge out of the eggs, at first they have a yolk sac. And so they can swim around, they don't need to feed. They kind of carry their food with them. Kind of like people leaving Brown Hall. But then, once you use up all that food you've carried out of Brown Hall, you got to find some resources. You start feeding. You start looking around for food. Some of you are taking mycology. You're looking for that chicken of the world. You're going down to the botans. You're going there. So, this to me suggests that there's some pretty intense competition among trout. And so, as y'all know, Thomas Malthus, right? More young are produced than can survive in the animal world. Humans, it's weird. But, so anyway, looking at this, it's interesting. You think about the importance of biotic versus abiotic factors in streams, of equilibrium versus disequilibrium forces. I look at this, I think if I'm a trout from year to year, the amount of resources in that stream are going to play a pretty big role on if I'm able to survive or not, Okay, cool. Mary Parr. So a lot of research that she did is kind of important to our understanding of roles of biotic forces in streams. So after doing research at University of Oklahoma, she moved to Cal Berkeley and did an experiment in the Eel River in California, which was published in the journal Science, which is about the highest scientific journal there is, which means that it really showed us something that we didn't know. It showed us something we hadn't known before. So here's the food web in the Eel River. Down in our first trophic level, we've got some diatoms. This is a filamentous green algae. This is like a blue-green algae. We got these going on. Then we've got chironomids, the larvae. Remember what those look like? They look like I don't know, like most dipterans look. Little worms, tiny little wormies. It's a technical term. So those algae were fed on by chironomids. Then we had some predatory insects. These are damselfly larvae. Roach fry. So a roach is it's a fish in California. These are not aquatic cockroaches, which would be slightly terrifying. Then we had stickleback, which is another fish. But notice they're the juveniles. So fry just means the young. What did you say? The bibbies. I haven't watched Z Frank stuff in a while. I don't know. We've been in different places lately, kind of mentally. And then up above there, we have some larger fish. So steelhead are fish that have metal heads. They're all into Dawkin and Metallica. Is Metallica even? They're not. I don't know. They sold out. Anyway. So, no, steelhead are rainbow trout that move back and forth from the ocean. And they're silvery, so they look like they got a steel head. That's why we call them steelhead. And then large roach. So these large. So we've got basically a 4 trophic level food web. And to make matters confusing, these large roach, sometimes they eat algae. They can't make up their mind. Sometimes they want a salad, sometimes they want meat. You know how it is. So this food web was going on there. So what Power did was she excluded this top trophic level, and then allowed those food webs to develop in the presence and absence of these adult fish. So, putting your HSS Fretwell brains to work, what would you expect to happen to this food web when these larger fish are excluded. We can start from the top down, like HSS, like they want us to. So if we take the top level of the food web, lop it off. So what's going to limit production of these dudes and dudettes? Exactly. Yep, exactly. So when these don't have predators eating them, resources limit them. Okay. If they're resource limited, then these midge larvae should be getting hammered pretty hard, because these populations here are allowed to increase as big as they can based on their resources. And so then, since there's less of these, you'd expect there to be a lot more production, right? All right. So if we look at the algal standing crop, so these exclosures were without these. So, sorry, this is at the beginning. This is the end. We want to look at the end of the experiment. So, does this follow the predictions of HSS fretful trophic theory? After 5 weeks, we've got our exclosures and our enclosures. What do you think? Yes, maybe. Yeah, so there's more algae in the exclosures, which is what we predicted, right? If we keep these away, we would predict that we would get more algae. Okay, so that matches up. This might sound kind of funny now, but this is a huge deal in streams. We hadn't ever seen this before. Demonstrated experimentally that top predators could have this big of an effect. Pretty significant. So the predators decreased chironomid populations. This is a lested damselfly. Terrifying, absolutely terrifying. She sampled the midge larvae. It's kind of cool when you come up with new experimental approaches, which is kind of like what we did with our sediment samples. Like, I don't know, like let's just do it. It makes sense. Let's do it. Like the whole thing we came up with about, y'all came up with, I just stood back and be like, yeah, that makes sense. About like taking the sieve off, then like pouring it through in the bucket, and then pouring that bucket through the smaller sieve. Like, that was brilliant. The way that she measured midge larvae was they were in the algae. Picking them tiny midges out of algae with forceps is a good way to decide to do something else with your life. So she had the algae, she scraped it off, put it in the bucket, just swirled it around like crazy. The midges came off. They're like, I don't know what's going on. Shh, I'm out of the algae. And then she put them through a sieve. It was pretty interesting. So yeah, the number of midges per bucket significantly higher in the control than those that had the predators around. So it looks like they were knocking down chironomid numbers. So the damselfly is scary. That damselfly is like the equivalent of that. Like if you were a midge larva, that would be the damselfly looking like to you. Or perhaps this is what would be scary. You know what that was, right? That was actually something we didn't see. We didn't see any hellgrammites, unfortunately. We didn't see any when we were messing around either. You have to see it again. And it's crawling around. It's kind of terrifying. Really? Oh, in the nets, right? Not in the, yeah. Yeah. So they're pretty terrifying. Also, if you Google hellgrammite, you know, they've got all these cartoon characters named after real predators just to be tough and stuff. But this is what the adults look like. And it is exactly, yeah, right. Sorry. So hellgrammites are Dobson fly larvae, but in the adults, they don't feed. These are just mate attractant pinchers. I don't know. I guess they look attractive or something. So hellgrammites are actually fairly intolerant. So yeah, so when you see them, it's a good indicator. All right. So that talks a little bit about the role of predation in food webs and how energy moves between trophic levels. This is a study where they looked at water striders, the family Garridae, the hemipterans. And they looked at water strider habitat use in pools with and without trout. They looked at the percentage of Garrod populations in the middle of the pool. And when there were trout in the pool, wherever more of the Garrods hanging out, in the middle of the pool or not middle of the pool? In the not middle of the pool. So this is an example of something we'll talk about in just a second with a paper discussion of predators not just influencing prey abundance and energy flow, but having more indirect effects and affecting the behavior of the prey, which can be important. This is another study from New Zealand where there's terraphyton and then a heptageneid mayfly that was consumed by galaxiids and trout. So galaxiids are a family of fishes that are native only to Australia and New Zealand. You know, kind of like their terrestrial species are just weird because like it drifted away from the rest of Asia a long time ago. Freshwater stuff is weird too. So galaxiids, they kind of look like little trout. But now trout have been introduced because people like to catch trout. So there's been a lot of research done on like how are these trout affecting the aquatic ecosystems. Okay, so they had an experimental setup, one that had no fish, one that had the galaxiids, one that had trout. And the height of these bars are essentially mass of chlorophyll. So here we have chlorophyll concentrations, actually the same units that we'll end up using for our study. And over here, they had ash-free dry mass, so like the biomass of the algae. Okay, and so then they had some treatments with no fish, some with galaxiids, some with trout. So if you look at those data, what might you conclude about the role of these predators on affecting the productivity in the system? So this is kind of an HSS Fretwell thing, where we've got our 3 trophic levels. We've got a predator, but maybe all predators are not created equal. What might you conclude from these data? Yeah, so so this is from the top of the stones, and this is from the sides of the stones. So if we just look at what was going on the top of the stones, what was happening? Yeah, something, for whatever reason, yeah, so we don't have data on the inverts, but there was definitely less algal production when you didn't have fish. Presumably either the inverts were being eaten or they're getting chased away. But what about on the sides of those? Yeah, so we've got trout here. Which of those predators has the strongest impact on those invertebrates? The trout, right? So both species eliminated the invertebrates, it looks like, from the tops of the rocks. But only the trout had a strong enough effect to actually reduce and eliminate those invertebrates from the sides of the rocks as well. So this is a non-native predator that's coming in that looks like it has a stronger effect on the ecosystem than the other species. So HSS Fretwell sort of takes this approach that, you know, it's just a trophic level is a trophic level. But sometimes species differ in how they operate. You kind of have to break that down. Yeah, and so there was in fact a significant difference here between the trout and the galaxiids on the sides. And so we asked, why did paraphyte increase, whether it was because they ate them or because it was a behavioral response. And in fact, this is the average number of these observed on the 8 cobbles with different predatory fishes. So not the number being eaten, but just the number that were on the surfaces. And so in fact, it looks like there was a behavioral effect where these invertebrates were able to sense the trout being around. And they've done studies that they're able to sense presence of other species by chemicals in the water. So if they're a trout, and it's interesting, right? Because in some ways, it's like, how do they know, like galaxids versus trout? Well, somehow they do. Or perhaps it's a visual response. Hard to say. Authors didn't investigate that. But there's a behavioral response as well. So this is something that was really learned about in the 80s and 90s. The impact of these non-lethal effects. So just the threat of predation can have really strong impacts on communities and can affect the way whole ecosystems work. Not because prey are consumed, but just because prey are responding behaviorally to the threat of a predator. So if we were to just tie up a couple things here, predation can affect prey by altering their habitat use and where they move around. They can reduce the population locally where there are scarce resources, in the example of the trout with Allen's paradox. And as we'll talk about in our paper discussion of the day, for things to matter evolutionarily, there's going to have to be an impact on fitness, right? So to evolve in response to predation, those that are able to evolve in response are going to have increased fitness. Meaning, how do we define fitness in an evolutionary context? Yeah, the ability to leave more copies of your genes in the next generations, essentially, right? So ultimately, there's going to be selection for species, individuals that are able to avoid predation, whatever that might look like. So, so these are different ways that predation can affect prey, and this can lead to these different types of ecosystem effects.